Good afternoon, ladies and gentlemen. It is a great pleasure for me to make a speech here. I'd like to thank the organizing committee for giving me this opportunity to communicate with you guys. The topic of my report is the repair of infected bone defect with Clindamycin-Tetrahedral DNA nanostructure. Antimicrobial resistance is a serious global health problem. A study jointly published by global multi-center researchers in authoritative journal Nascent showed that the deaths caused by AMR exceeded many recognized causes of death. In 2019, 1.27 million people died directly from AMR, and 4.95 million deaths were related to AMR infection. The AMR problem has become an urgent problem to be solved globally. Infectious bone defect is a worldwide problem due to the coexistence of bone infection and bone defect, difficult treatment, long course of disease, high surgical failure rate, and high disability rate. Currently, biological materials used in clinical practice have poor osteogenic induction and in vivo degradation performance. Dead bone without blood transport and most interlock fixation materials are easy to cause bacterial attachment and force highly structural and adhesive bacterial biofilms, which are difficult for antibiotics to reach and form a high concentration environment. Dense biofilms greatly inhibit and killing effect of antibiotics on bacteria. It can then lead to the drug-resistant bacterial infections. Biofilm-forming bacteria are 500 to 5,000 times more resistant to antibiotics than normal bacteria, including those that are normally sensitive to antibiotics. When the bone defect area is infected, it will also lead to local blood supply deficiency, lack of osteoblast, decrease of bone regeneration ability, and many other factors that hinder bone formation. The spread of infection and necrotic tissue, as well as the lack of local bone-inducing factors, may also delay the repair of bone defects, leading to the poor therapeutic effect. How to safely and effectively control the infection of bone-resistant bacteria and repair the bone defect is a bottleneck problem to be solved at present, but also an important research hard part. Although breakthrough has been made in the research and development of tissue-engineered bone, and in recent years, there are still limitations in how effectively control the progression of infectious bone defect lesions and achieve induced reconstruction and functional repair of bone defects. Therefore, it is of great clinical and scientific significance to develop a biological scaffold material with high antibacterial efficiency, promoting local angiogenesis and reducing our bone formation. The commonly used methods to combat bacterial biofilm, including nano-coating that inhibit adhesion, surgical treatment combined with antibiotics, and liposome nanoparticles. With the rapid development of nanoparticles in recent years, researchers have introduced selectively targeted biofilm substrates to enhance the specificity of nanoparticles and thus improve the efficiency. However, currently used nanoparticles have strong cytotoxicity, affecting the normal physiological activity of host cells and poor biocompatibility. Tetrahedral DNA nanostructure is a nanostructure self-assembled by 4 single-strand DNA through precise base pairing, which is highly editable. DNA has good biocompatibility and strong affinity for bacteria and mammalian cells. Our research group have carried out in-depth study on TDNs in the early stage and confirmed that the TDN has anti-inflammatory and antioxidant property, can promote the proliferation and differentiation of animal cells, and its unique nanostructure makes it an efficient carrier for drug entry into cells. It has shown unique advantages in targeted therapy, anti-therapeutic, anti-tumor, coronary regeneration, and osteoarthritis treatment, and many other aspects. The size and structure of TDN can be precisely regulated. By using TDN as a carrier to deliver ampicillin, the two of them can form a tight and stable complexes, thanks to TDN's affinity with bacteria and mammalian cells. The compound will deliver more ampicillin to the surface of muscle, and compared with ampicillin alone, the MIC was significantly decreased. Morphological examination showed that the structural the complex damage in the external morphology of muscle more than ampicillin alone. Our previous study have confirmed that TDN can combat oxidative stress and alleviate astrocyte apoptosis. In addition, it can promote the differentiation of astrocytes from pro-inflammatory A1 phenotype to neuroprotective A2 phenotype, thereby reducing the size of cerebral infarction caused by a transient middle cerebral artery conclusion and promoting the recovery of nerve function. In addition, TDN can protect the astrocyte in stroke by downregulating the Taurus receptors and NF-κB signal pathway. This study provided a new strategy for the treatment of ischemic stroke, and relevant results have been published in an authoritative international journal. Hydrogels are beneficial to the migration and growth of cells in the process of tissue regeneration and wound healing due to their biomimic property similar to extracellular matrix, highly three-dimensional hydration network structure, and powerful ability of nutrition delivery. They are very suitable to be used as a carrier for the slow release of transplanted cells or growth factors to accelerate tissue engineering. Geoma is a photosensitive hydrogel material with excellent biocompatibility. It can also simulate curing reactions by ultraviolet light and visible light to form three-dimensional structure with certain strengths suitable for cell growth and differentiation. Its biocompatibility is much better than that of matrix glue and fabric glue, and its properties are similar to collagen. At the same time, the formability is much better than collagens, which is the best choice to replace this kind of materials. BMSA are good osteogenic differentiation ability, although the osteogenic activity of porous scaffold is relatively clear. Due to the limitation of traditional production technology, how to accurately control the internal porosity, pore size, and interpore traffic structure of biological materials has become the bottleneck limiting its development. The development of two aqueous biological ink provides new ideas to solve these challenges. Composed of immersible phase geoma and PO, this biological ink can be directly photosolidified to form hydrogel structure. Furthermore, a porous scaffold can be formed by removing the PO phase after optical cross-linking gel. And the aperture of the scaffold can be controlled by adjusting the ratio of the geoma and PO phase. Compared with traditional biological scaffold, geoma PO hydrogels have better biocompatibility, cell proliferation ability, and metabolic ability. Meanwhile, the antibacterial properties and osteogenic ability of this porous hydrogel can be enhanced by a combination of drug-loaded nanomaterials and osteogenic induction factor. 3D bioprinting takes living cells, bioactive factors, and biomedical materials as basic forming unit to design biological active artificial organs, implants, or cell three-dimensional structures. It is an emerging discipline of the cross-integration of medical science and manufacturing science, and it is the most cutting-edge research field of 3D printing technology at present. It is also the most dynamic and promising directions of 3D printing technology. The development of 3D bioprinting technology makes it possible to accurately design and prepare porous structure of biological materials. At the same time, it also makes it possible to design personalized medical implant material. In theory, 3D printed drawing of personalized bone repair materials, called CAD models, can be obtained only from 3D scans data of the experimental animals. In view of the above relevant analysis and summary, combined with the previous research foundation of our group on TDN drug loading, 3D bioprinting, bone defect repair, and geoma PO porous hydrogel materials, this study aims to address the disciplinary problem and clinical bottlenecks of bacterial biofilm formation, antibiotic resistance, and bone tissue regeneration difficulties in the treatment of infectious bone defect. Through bone tissue engineering and 3D printing technology, this project aims to build an individualized slow-release biological scaffold that can be long-term controlled release, collaborate with high antibacterial agent, promote local angiogenesis, and induce bone regeneration. The optimal reconstruction of bone regeneration microenvironment was achieved from the micro nanoscale and three-dimensional spatial structure, effectively promoting the repair of infectious bone defect. The safety and effectiveness of sustained release system was verified through biological evaluation and animal experiment, providing complete preclinical data and provide a new strategy for treatment of infectious bone defect. In the first part, we designed an injectable gamma hydrogel to simulate the skeletal extracellular matrix. First, the physiochemical property of gamma hydrogel was determined by SEM, NMR spectrum, DMA, and viscosity test. Immunofluorescence results showed that the hydrogel scaffold had good biocompatibility. BMC proliferated well in the hydrogel scaffold. In vivo experiment, the gamma hydrogel scaffold loaded with BMC was implanted into a rat model of segmentary bone defect to verify its bone repair ability. The morphology and mechanical property of gamma hydrogel groups loaded with BMSA were significantly better than those of other groups. HE results showed that the gamma hydrogel scaffold loaded with BMSA can significantly promote the bone regeneration and angiogenesis compared with other control groups, which is the ideal synthetic substitute. In the second part, we upgraded and optimized the hydrogel material compared with Gamma PO BioQuirs BioInk and added NHA to prepare a hydrogel scaffold with similar mechanical properties and microstructure to natural bone. It is a 3D scaffold with gradient NHA to assimilate and different components of articular cartilage, subchondral bone, and bone marrow. We tested the characteristic of gamma PO hydrogels in different properties, and as well as NHA propagation. This is the surface and cross-section of NHA gradient bracket. With the change of gamma PO ratio, the internal pore structure of hydrogel shows different sizes. In the 1,3 and 1,4 group, the continuous phase cannot be maintained, and photocuring cannot be achieved by UV evidation. In the 3D reconstruction image, it can be observed that the 1,1 group can better realize the connected pore structure. Subsequently, SEM biochemical and rheological tests were used to further clarify the characterization of hydrogel scaffold. In the same state of the in vitro culture, porous hydrogel showed higher cell viability and cell proliferation ability, among which 1,1 group was the most significant. The effect of porous structure on cell morphology and was observed by cytoskeleton staining. The results showed that the interconnected pore structure of 1,1 group could better promote the cell stretch and the formation of the intercellular connections, which is one of the most important characteristics of hydrogel scaffold. Ultramarine red staining was used to investigate the osteogenic differentiation ability of hydrogel-coated BMSA. Calcium nodule and NHA particles formed by BMSA in the upper and bottom layer of BMSA scaffold were red. Expression of osteoblast genes and hydrogel-coated cells were quantitatively analyzed by qPCR. The results showed that the porous structure could promote the expression of osteo-related genes like OPN, ALP, VEGF, while the dense non-porous structure was conducive to the expression of chondrogenic cells like AGG, SOX9, collagen X. In vivo, the results showed that the cell-loaded scaffold with gradient NHA and pore structure can promote the overall repair of cardiac, subchondral bone, and bone tubuli. In the third part, on the basis of previous study, we used the 3D bioprinting technology to construct a biological scaffold with both efficient antibacterial and osteogenic induction compatibility to solve the problem of antibiotic resistance and bone tissue regeneration difficulties in the treatment of infectious bone defect. We first synthesized the TDN by a self-assembly and then loaded the clindamycin on the TDN by electronic absorption. The TDN clindamycin composite structure was characterized by Page, TEM, and atom force microscopy, zeta potential, and other means. Immunofluorescence results showed that TDN could effectively transport antibacterial drugs into cells, and the CCK8 results showed that the TDN clindamycin component structure could effectively promote the proliferation of BMSC. In vitro antibacterial experiment confirmed that TDN clindamycin composite structure can significantly reduce the MIC of clindamycin and significantly improve the antibacterial performance. Further, we constructed a bionic composite scaffold loaded with TDN-CLI composite structure, porous hydrogel, and BMSC by 3D bioprinting technology. The bionic scaffold loaded with TDN-CLI can achieve long-term and stable release of clindamycin. The results of immunofluorescence showed that the composite scaffold had good biocompatibility and cell stretch. In addition, the results of SEM and osteoinduction experiment showed that the antibiotic effect and the effective induction effect of composite scaffold were significantly better than those of other groups. These results demonstrated that the theoretical feasibility of 3D printed TDN clindamycin biotic scaffolds in controlling infection and promoting the repair of infected bone defect. Next, we constructed an infectious bone defect model of SD rats and implanted the composite scaffold. The in vivo osteogenic and antibacterial properties of bionic composite scaffold loaded with TDN-CLA were further evaluated by microCT three-dimensional reconstruction. The quantitative analysis, GIMSA and tunneling blue staining, HEM and Mason staining. The results showed that the bionic composite scaffold loaded with TDN clindamycin could release the antibiotic quickly in early stage, effectively control the infection, inhibit osteoclast, promote osteogenesis, and further promote the repair of infectious bone defect. Immunohistochemical analysis of infected bone defect specimens in each group showed that the expression of bone-related genes of composite scaffold was significantly better than that of other groups. The study concluded that the infectious bone defects are difficult to repair and may lead to antibiotic resistance. TDN could provide an efficient drug delivery carrier. The GEOMA scaffold can be used as a cell carrier of PMSA to promote the repair of segmental bone defects and has great potential in future clinical applications. The TDN clindamycin molded hybrid scaffold developed in the current study has broad application prospect for treating infected bone defects. Well, that's all of my statement. Thank you for your attention.